So this is common knowledge about hyperthermia. We use the capacitive type developed by the Oncotherm limited company with whom we collaborate in a grant. As you probably all know, modulated electro-hyperthermia induces membrane perturbation, and cancer cells selectively observe the energy. And the destruction of cancer cells leads to immune activation. We study triple-negative breast cancer because of its clinical relevance. Without any receptors, there is no targeted therapy, and therefore there is much worse overall survival in these patients. So there is a sogenic cancer model for triple-negative breast cancer in biopsy mice, and this is the 41 model. And we have isogenic clones with different metastatic potential of the same cell line, and they have different epithelial, mesenchymal, and mesenchymal epithelial potential, and only the 41 is able to perform macrometastasis, whereas the 4007 can also form spontaneous micrometastasis, but without MET, they don't form big metastasis. We investigated these cell lines after implanting them into the mice and found that the 41 cells have an immune desert phenotype, much less immune infiltration than the 4007. And also we found that two cell lines have a high expression of immune checkpoint inhibitor molecules like the CTLA-4, the PD-1, and the PD-1 ligand. But the 41 has a much lower expression. Within this grant, we participated in improving the previously used Labahi used for treatment of mouse tumors. The tissue electrode was replaced with this columnar electrode. Much more accurate coupling is achieved, and the instrument has also been developed. So instead of less reliable heating, we inserted 4 electrodes into the rectum, underneath the skin, next to the tumor, and into the tumor, and we could reproducibly provide a 2.5 degrees higher increase in temperature in the tumor. So we injected 10 to the 6 41 or 4007 triple-negative breast cancers into the forced memory fat pad, and after the inoculation, the tumors were grown for 6 days, and then with the traditional caliper and ultrasound methods, we measured the tumors and randomized the mice into sham or Labahi 200 treatment arms, and the mice were treated either every 48 hours or every 72 hours, twice or 3 times. And in the 4007, we also have data from 5 treatments. So regarding the traditional tumor measurement, we didn't find an effect of the treatment after 2 treatments, and after 3 treatments, there was only an effect if the tumor was left to grow long enough, and the substantial exponential phase tumor growth was inhibited when the treatments were performed every 72 hours. However, despite the lack of measurable tumor size growth, we could detect in fluorescently labeled tumors a significant reduction of viable tumor cells after 2 treatments. And on the histology, we could find a large tumor destruction already after 2 treatments in the treated mice, but not in the control ones. Although the tumor size is similar, there is a big destroyed area in the middle. And we also performed HSP70 staining. You can see here the necrotic core and surrounded by HSP positivity. And we regarded this pale staining as background. And we could see that the modulated electro-hyperthermia treated tumors had much more specific HSP70 staining. The tissue destruction ratio was elevated in the 41 cells after two treatments. This was quite similar after 2, irrespective of the treatment intervals, whether it was 48 or 72 hours. Also, the HSP70 was upregulated after 2 treatments in 41, but this was again similar in 48 or 72-hour treatments. All the mice were losing weight during the treatment, and this weight loss was more expressed if the treatment intervals were longer, and also the weight loss was more expressed if the treatment was active compared to the sham. Comparing the 41 and 4007 cell types after 3 treatments, there was no significant difference. However, interestingly, in the 4007 animals, there was a tendential increase in tumor size, whereas in the 41 there was a tendential decrease. And comparing 3 treatments in the 4007 and the 41, we see that the red lines are the treated mice and the gray lines are the sham mice. And there is no significant difference, however, a tendential increase in the 4007, whereas there is a decrease. The asterisks are missing here, but there is a significant decrease, and also after removal of the tumors, the tumor weight is smaller in the 41 cell lines. So the similar treatment protocol seems to be a little bit more effective in the fast-growing immune desert tumor type. Also in this case, despite of the no decrease in tumor size, we see increase in tumor destruction as well as HSP70 expression. Here is a comparison of 3 treatments in the 4007 and 5 treatments in the 4007. And again, you can see that there is a slight increase after 3 treatments and no significant difference in the tumor weight. But even in this tumor type, after 5 treatments, we see a significant decrease in tumor growth and also tumor weight. Here the average body weight loss is again different between the 41 and 4007. 41 mice lost more weight, especially in the active arm, suggesting that tumor destruction was inducing immune activation, which is a probable explanation why these mice were losing weight. We also checked the immune infiltration, but we only have data from, we only have very preliminary data on this. All the CD4, CD8, and macrophage marker were actually reduced in the hyperthermia treated mice. But this was performed just 24 hours after the last treatment. So we believe that the treatment itself was devastating on the immune cells, but we hypothesized that if we wait longer, then we will see an actual immune activation. Unfortunately, we don't have that data yet. So in conclusion, even a single treatment, I didn't show, was able to increase tumor destruction. This effect manifested in measurable tumor size reduction only after 3 treatments with 72-hour intervals. So this seems to be a more effective and less stressful for the animal, as we could see from the body weight changes. Moreover, our preliminary data suggests that modulated electro-hyperthermia was more effective against fast-progressing 41 compared to slow-progressing 4007. And next we will investigate if modulated electro-hyperthermia can immune sensitize the 41 for immune therapy checkpoint inhibitors. And I would like to thank you with my colleagues. Later today, Forika Gertrud will give a talk on radiosensitivity induction by hyperthermia in mice, and we have 3 posters. Thank you.
Thank you for your presentation. Are there questions from the audience? Yes, sir.
You didn't mention the temperature you applied if you use modulated electro-hyperthermia. Is it a standardized treatment, or what temperature did you achieve, or did you measure the temperature?
Yes. We inserted 4 catheters in a preliminary pilot experiment and measured with the old machine. We could reach a temperature elevation of 1 to 2 degrees, but quite unconstantly with the Labehi 200, and also with the new electrode, we could achieve a constant 2 degrees temperature elevation from the tumor surrounding within the tumor. And then we recorded the settings, and we used these settings throughout all the experiments. We were controlling the temperature, but not intratumoral, because if we puncture the tumor, that can induce inflammation and tumor destruction. So we were measuring only the temperature in the tumor surrounding.
But the temperature was not high.
No, 42 degrees.
40 degrees or something.
Yeah.
Okay.
The first slide was very nice with the epithelial mesenchymal transition, and then the mesenchymal epithelial transition. What would happen if you would give your hyperthermia at the left side of your slide? Would you influence the epithelial mesenchymal transition process as such, changing your biology at the right side of the slide?
Well, on the slide there were different. On the slide there were different tumor cell types. They are isogenic clones of the same 41. They are 41 derived mutants with different metastatic potential. We only investigated those which are capable of leaving the primary tumor, so which are capable of the EMT. We didn't investigate the 67 Farn tumor type, which we have, and it's not capable of EMT. So I'm not sure if hyperthermia is able to influence these processes, but I think it's an interesting question.
Unfortunately, I would really love to talk about the immunology of slides, but I think that would be something really new. Thank you for your presentation. And,